91,000 converts, 5 decades of migration, 6 generations of marriage inside the same community. When geneticists finally compared the DNA of Utah Mormons against the rest of the American population in 1984, they were not prepared for what came back. The DNA told a different story. Utah Mormons did not look like modern Americans. They looked like a 19th century Northern European village, frozen in time. To understand what those geneticists found, you need to understand what they were measuring. On one side, the descendants of roughly 91,000 European converts who left Britain, Denmark, Sweden, Norway, and Germany between 1840 and 1890 to gather in the Salt Lake Valley. 6 generations of farmers, weavers, miners, and shipwrights who crossed an ocean to follow a new religion into the high desert of the American West. On the other side, mainstream American DNA, the continuously mixing genome of a nation that absorbed Italians and Greeks after 1900, Eastern Europeans between the wars, Mexicans and Central Americans after 1945, East Asians and South Asians after 1965, and millions more from every continent in the decade since. 2 populations, same country, same passports, same supermarkets, but sequence them, and you are looking at 2 photographs taken 100 years apart. That contrast is the whole story this video will tell. One population sequence today reads like a snapshot of 1890. The other reads like a layered, modern composite. Same maps, same census forms. 2 completely different photographs at the molecular level. The first scientists to put the 2 populations side by side were Thomas Mellon, Lynn Jordan, and Mark Skolnik, working at the University of Utah. Their paper in the American Journal of Human Genetics, published in 1984, is the foundation of everything that came after. They measured gene frequencies across red cell antigen systems, electrophoretic markers, and the HLA-A and HLA-B loci, standard biology, standard methodology. They compared Utah Mormons against a United States reference sample, then against 13 separate European populations, then against 7 other religious isolates, the Amish, the Hutterites, the Mennonites. You would expect 2 things going in. First, you would expect Utah Mormons to look broadly American by 1984. 6 generations is a long time. People marry. Genes flow. Second, you would expect that if they did not look broadly American, they would look like the Amish or the Hutterites. Small, closed religious communities tend to drift genetically. A founder population of a few hundred always ends up looking strange after a century of marrying its cousins. Neither prediction came true. Utah Mormons did not look like 1984 Americans, and they did not look like the Amish. They looked like their Northern European ancestors almost exactly. Mellon, Jordan, and Skolnik wrote it plainly in the paper. The Mormon gene frequencies were similar to those of their Northern European ancestors. The reason had nothing to do with isolation. It had to do with 2 things: a very large founding population and a very large amount of incoming gene flow from one specific source. Translated out of academic English, that means too many founders to drift and a steady stream of new converts arriving from the same villages. The original founders had left. The Amish drifted. The Hutterites drifted. The Mennonites drifted. Utah Mormons did not. They kept the gene pool stable for 150 years because every new wagon train, every new sailing ship, every new Liverpool packet brought in the same kind of personal wagon train before it had brought Northern European Protestant working class people, very often from the same county. But the story gets stranger. In the 1980s, a geneticist at the University of Utah named Ray White was looking for a population to anchor what was becoming the world's first map of the human genome. He needed families, multi-generational families, large sibling groups, cooperative donors, meticulous record-keeping. Where in the 1980s, do you find 48 large, multi-generational Northern European families with documented pedigrees stretching back to specific 19th century European villages? You find them in Salt Lake County. Ray White collected 48 Mormon families from Utah and sent the samples to the Centre d'études du polymorphisme humain, CPH, in Paris. Those 48 families became the original international reference panel for human genetic variation. A decade later, When the HapMap project needed a reference group for Northern and Western Europeans, they used those same families. They named the group the CU population, Utah residents of Northern and Western European ancestry. When the 1000 Genomes project picked up the work, they used the same families again. If you have ever taken a DNA test from AncestryDNA, from 23andMe, from MyHeritage, and seen the words Northern European on your results page, those words are calibrated, in significant part, against Utah Mormons. Your DNA is being compared indirectly to 48 families in Salt Lake County. That phrase on your results, Northern and Western European, is the formal scientific label for the CU population. Behind the percentage is a small group of Utah Mormon families whose great-great-grandparents stepped off a sailing ship at New Orleans or New York more than 150 years ago. The reference standard for the entire Northern European world is a single county in Utah. That makes the next question matter. Who exactly were those 48 families descended from? Start with the British. Roughly 52,000 converts left the British Isles for Utah between 1840 and 1890. Most were working class. Lancashire mill workers from Preston, Liverpool dock laborers, Welsh coal miners, Scottish land farmers, West Midlands weavers, and shoemakers. The first organized immigrant ship was the Britannia, June 6, 1840. 41 converts from Preston under a leader named John Moon, sailing out of Liverpool. By 1845, fully 1 in 4 people in Nauvoo, Illinois, the Mormon headquarters at the time, had been born in the British Isles. 1 in 4, 5 years after a single ship. That single voyage started what church historians later called the Preston Branch, a working-class Lancashire congregation that became the launching pad of the entire British mission.
30 years later, Preston was still sending more converts to Utah than any other town in England. Liverpool became the operational center of Mormon immigration for the next half-century. Over 80% of all Mormon immigrant departures from Europe came through that one port on the River Mersey. If you have Utah Mormon ancestry, the odds are overwhelming that one of your great-great-grandfathers walked up the gangplank at Liverpool with a tin trunk and a ticket paid for him by a man he had never met. That single number, 80%, is half the reason Utah Mormon genealogies are so clean. Almost every line of descent runs back through the same English port, the same shipping company, the same Mormon migration officer who recorded each passenger's name, age, parish of origin, and date of arrival. The records exist because the migration was institutional, not accidental. Then come the Danes. Roughly 22,000 to 24,000 Danish converts followed the British across the Atlantic. They were organized by a Mormon mission led by Erastus Snow, working out of Copenhagen. The Danes did not scatter. They concentrated Sanpete County in central Utah and Sevier County just to the south. These two valleys became Danish strongholds so dense that Danish was still being spoken in some Mormon congregations into the early 20th century. Walk through the headstones of Ephraim, Manti, or Spring City today, and you will think you have wandered into Jutland. Next, the Swedes. A smaller stream than the Danes, but still substantial. The Swedes settled mostly in northern Utah, Cache County, Box Elder County, and the land around Logan and Brigham City. Lutheran villages from Scania and Östergötland transplanted to the foothills of the Wasatch Range. Then the Norwegians, the smallest of the major Scandinavian contingents, around 3,400. The first wave came through the older Norwegian colony at Fox River, Illinois. The most famous of them, Ellen Sanders Kimball, was born in Telemark in southern Norway. She walked into the Salt Lake Valley with the very first pioneer company on July 24, 1847. And finally, the founding pioneer aristocracy. These were the original handful, the early Anglo-American converts from New England, from upstate New York, from the Upper South, who joined the church in the 1830s, marched across Missouri and Illinois, and arrived in Utah before the European missions had even fully opened. Brigham Young was born in Vermont in 1801. Heber Kimball, Vermont, 1801. Wilford Woodruff, Connecticut, 1807. Joseph Smith, the church's founder, Vermont, 1805. The original Anglo-American core of the religion came almost entirely from one corner of New England in a single generation. When those founders gathered in Utah, they brought the Puritan genome of Upper New England with them. A single one of them, multiplied by plural marriage and 150 years of descent, can account for thousands of living Utah Mormons today. 5 streams: British, Danish, Swedish, Norwegian, and the Anglo-American founding stock. That is the genetic backbone of the modern Utah Mormon population. So why did these 5 streams stay so tightly held together? Why did they not dissolve into the broader American population the way every other immigrant group eventually did? 3 reasons. First, the Perpetual Emigration Fund. Brigham Young established it in 1849. It was a revolving loan fund that paid the transatlantic passage of impoverished European converts in exchange for repayment after they had settled in Utah. It institutionalized the migration, and it also narrowed it. The fund did not bring in random Europeans. It brought in the specific working-class Protestant communities where Mormon missionaries had built congregations, the same villages, the same trades, the same religious profile. Second, the migration corridor itself. Until 1855, every Mormon immigrant from Europe traveled the same route: Liverpool, New Orleans, up the Mississippi to St. Louis, across Iowa and Nebraska, and then by wagon or handcart over the Rocky Mountains to the Salt Lake Valley. After 1855, the route shifted to Liverpool, New York, and the railroad heading west. Scandinavian converts funneled through Copenhagen, Kiel, Hamburg, and Hull on the English coast before arriving in Liverpool, where the Mormon European headquarters was located. Ask a Utah cousin which port his great-grandfather sailed from. The answer, 9 times out of 10, Liverpool. Third, the cutoff. In 1896, Utah became a state. The church wound down its policy of gathering converts to Zion. After 1890, the genetic doors largely closed. New converts stayed in their home countries. Existing Utah families married within the community. The Utah Genome stopped absorbing new ancestry at exactly the moment the rest of America was about to be reshaped by the great immigration waves of the 20th century. The mainstream American genome kept mixing. The Utah Mormon genome held its 1890 shape. That 1890 shape, preserved with such clarity, turned out to be a gold mine for medicine. In the mid-1970s, the University of Utah began linking Mormon pioneer genealogies into a single computerized database, the Utah Population Database, or UPDB. It is now housed at the Huntsman Cancer Institute. It contains more than 11 million linked individuals across as many as 17 generations, seeded from roughly 170,000 founding families whose ancestors had a birth, marriage, or death somewhere along the Mormon Pioneer Trail, or in Utah itself. Nothing like it exists anywhere else on Earth. Researchers can pull any single founding ancestor and chase his or her DNA forward through every descendant the database contains. They can compare healthy descendants to sick ones across 6 generations. They can isolate the chromosome region where a disease mutation must lie. No university in Europe has anything like this. No university in Asia does either. The combination of Mormon record-keeping, 6 generations of stable population, and modern computing power is unique on Earth. When Mary Claire King's laboratory localized the BRCA1 breast cancer gene to chromosome 17 in 1990, the race was on to clone it. The team that finally did so in 1994 was led by Mark Skolnik, the same Mark Skolnik from the 1984 comparison paper. He used extended Utah Mormon families pulled from the UPDB. The breast cancer gene that has now been tested in millions of women was first read out of pioneer pedigrees. BRCA2 followed in 1995. It was sequenced by Skolnik's company, Myriad Genetics, same archive. The APC gene responsible for familial colon cancer was tracked through the descendants of a single pioneer named Lyman Hinman. Genes linked to melanoma, prostate cancer, cardiac arrhythmia, and long QT syndrome were all first mapped through Mormon descent groups in which one 19th-century ancestor could have hundreds of traceable descendants. Ray Gesteland, who served as vice president for research at the University of Utah, said it plainly in 2008, "More human disease genes have been discovered in Utah than in any other place in the world." One distinction matters before we close. The phrase Utah Mormon DNA refers to a specific historical population, the descendants of those 19th-century European converts, 6 generations deep in the Salt Lake Valley. It is not the same thing as Latter-day Saint membership today. Modern membership in the Church of Jesus Christ of Latter-day Saints is over 17 million people worldwide. Most of those members live outside the United States. Increasingly, they are Latin American, Pacific Islander, African, and Asian. Millions of contemporary Mormons share none of the Northern European founder signature this video has been describing. They are members of the same faith. They are not members of the same genetic cluster. What the geneticists found in Salt Lake County was a demographic accident, a specific 50-year migration window from a specific religious mission that produced a specific, frozen snapshot. Nothing more, nothing less. The Amish drifted. The Hutterites drifted. The Mennonites drifted. Utah Mormons did not, because the wagons kept coming. Because the Perpetual Emigration Fund kept paying the passage. Because the Liverpool packet kept sailing. Because the same 5 streams of Northern European villagers kept flowing into the same desert valley until, in 1890, the doors quietly closed and the genome was set. 6 generations later, when scientists wanted to know what Northern European DNA actually looked like, they did not go to Northern Europe. They went to Utah. A people can move 2,000 miles and still carry their village in their blood. Next time on Anglo Origins, we leave the desert and head east. The Pennsylvania Dutch arrived in America a full century before the Mormon pioneers. Their DNA tells an even stranger story.